It's a deeply personal decision that must be made by every doctor, researcher, and scientist working in stem cell and fetal tissue research. Where do I stand on the moral and ethical questions about conception, life, and human dignity? For some, science trumps all. If a single person can be saved, research should never be limited. The end justifies the means. But others wrestle with the gut-wrenching feeling that one human life is being sacrificed to save another. Some of the best and brightest minds in the world fall on opposite sides of this debate, while dedicating their lives to finding cures and ending suffering. Here's one researcher's journey to her decision.
When I was directing a research lab and I was studying, again, congenital heart disease in babies, and whenever I would apply for grants, they kept coming back to me and saying, you need to use cell lines that are specifically human. And they kept pointing me to these human cell lines that have been isolated from babies that have been aborted years ago. And that really started some serious soul searching for me. I mean, if we look at the advancements that have been made over the years and the number of babies that are being saved now at an earlier stage, I mean, the age of viability has been moved back. The clock is being moved back. Babies as young as 21 weeks have survived with active care. And we see that babies as early as 15 weeks have actually been saved because of advanced fetal surgery. Why would we continue to do fetal tissue research from aborted babies and destroy human embryos for research and for medical advancements when those same babies can be treated inside the womb and can be saved? And so then in my own research, I realized, wait, there was an awesome alternative that I could actually work with the surgeons. And when these babies underwent heart surgery, they would discard some of their heart valve tissue that they didn't need anymore. And I'm like, and to me, it was incredibly valuable. So I would be in the OR waiting for them to call me. I would then collect that discarded tissue. We took it back to the laboratory and we were able to isolate some really important cell lines that really transformed our work. And I learned this technique in Boston at Harvard Medical School. And then I carried that into my laboratory at the Medical College of Wisconsin, where we actually developed diagnostic tests for children to try to understand the genetic cause of their disease.